Oh. All right, so I guess we can start here. Let me just get this. Out of the way. So I'm going to talk about a couple of different tools today. It's useless. BioNetGen is the software framework that we've developed for doing rule-based modeling. It's basically a language and a set of software simulation tools that for interpreting the language. Oh. Yeah, it's fine. All right. It's good for this. And RuleBender is the graphical interface for using for using those tools. It's really more of a programming environment than it is a pure graphical interface for the tools. But and we'll be demonstrating those a little bit after the break, and Jose will lead you through sort of a hands-on tutorial so that you get a handle on making the reaction networks in that software. So what I wanted to do first was just to give a little bit of background. So you know about the BTRC. No, it's now called BTRR. I have to update my slides. We're a biomedical technology research resource. And so the tools that we're developing, which include BioNetGen and RuleBender and MCell and CellBlender and Cell Organizer and so on and so forth, are all being developed under the auspices of this center, which is funded by the NIH. And part of our activities include giving workshops like this to disseminate the tools to the research community. So this is a big part of what we do. And the multi-scale aspect of this is really critical. So we want to, I think Marcus showed this slide before. I forgot how busy it was. But all I want to show is that the goal here is of the overall center is to go from the molecular level. So one research component of the center involves development of tools for molecular modeling and simulation, and also visualization. The middle level component of this, the research resource, involves development of spatial and biochemical simulation tools for complex systems, and this is a snapshot of the movie that Tom showed you yesterday. And then finally, at a higher level, reconstructing complex neural circuits at even a higher spatial scale, so how cells interact with each other and how their interactions affect signal processing and behavior at the highest level. So the overall goal is to eventually connect all of these different levels, and we already even talked about different kinds of projects, modeling myocytes in the context of heart physiology, and modeling neurons in the context of some complex neural network. Those are examples where we need to understand proteins at the molecular level, build those into models that describe dynamics on the cellular level, and ultimately how cell-cell interactions couple to those. Okay, so enough is enough with that. From an organizational perspective, these are the sort of three technology components going from the molecular, cellular, to tissues scale, and we're 2/3 of this workshop is basically the TR&D2 component, the subcellular to cellular modeling. That's MCell and BioNetGen and so forth. At this higher level of considering whole cell and beyond is this TR&D3, and the part about the cell organizer part is actually Bob Murphy is part of the TR&D3 component. And we have a lot of different kinds of biological projects that are motivating this. The development of our tools. I just wanted to mention, we talked yesterday, I think, about different modeling approaches for example, handling spatial complexity, going from continuum approaches where you have coupled partial differential equations. Mesoscopic approaches are kind of in between, where you consider well-mixed subvolumes, and then all the way down to considering distinguishable molecules, which is what MCell is doing, where you consider the diffusion of individual particles. You could, for example, trace a particle in the simulation. And these are relevant at sort of different concentration in different concentration regimes. When you have very low concentrations and small numbers of particles, then noise effects are can have a strong effect on the dynamics. Similarly, when the complexity at the spatial scale is becoming comparable to the to the changes in concentration over your geometry, then also this kind of an approach becomes important. And another aspect of the complexity, so spatial resolution is one feature of the different simulation approaches that people have developed. Another is this ability to handle reaction network complexity, and that's what I'll talk about today, going from a situation where you write individual reactions in your system by hand to one where you have very complex molecular networks that form very dense numbers of possible states and reactions, and where you need different kinds of approaches for specifying those networks. So a motivating example in this case, and I could have pulled up some more details, but is this CAM kinase 2 molecule. So CAM kinase 2 exists as a dodecamer. So there are 12 subunits in the functional complex that's present at postsynaptic densities, and it couples multiple signals from different receptors in this postsynaptic region, and is involved in activating a whole host of downstream targets. So the complexity here is enormous. CAM kinase 2 is regulated by tyrosine phosphorylation at multiple sites. It's regulated by the binding of calmodulin, which is where it gets its name. It's calmodulin regulated kinase. And I was relying on Tom to provide the details because I've forgotten how many binding sites and so forth. But when you figure out how many different possible states there are for a single calmodulin subunit in this dodecamer, there are, depending on different levels of abstraction of modeling, approximately 40 states. So there are at least 2 tyrosine phosphorylation sites. There's a calmodulin binding site, and I think there are calcium binding sites. I can't remember what in detail the specific features of the regulation. We can go through that as an exercise later on. But the idea is that as soon as you start dealing with a molecule, a protein, which I'll represent here, which has multiple sites that can be modified, that can bind to other, for example, proteins. And here, I think part of the complexity comes from the calmodulin protein, which has multiple calcium binding sites. So you wind up with many different states, possible states of at each of these different interaction positions. So here there are 2 possible states. Here there are 2 possible states. Here there could be as many as 5, for example. And if the number of possible then states of the whole molecule is the product of these numbers, or 20 different possible states. You have a correction? I'm making this up, so I don't remember the details. It's just an illustration. But the point is you have interacting molecules that have multiple possible modification states, and this creates this exponential explosion in the possible number of complexes that you need to consider. So, I mean, it's possible to kind of coarse grain this, to say, okay, we're not really interested in modeling all 4 possible phosphorylation states of the CAMK2. We're going to just collapse that down into an active and inactive state. That might be adequate for many kinds of applications, but would not be adequate for trying to understand the effects of mutation at either one of those sites, for example. Is that making sense, or have I lost people with this? Good? Good, thank you.
I just had a random question. Is the structure, is the crystal structure for CAMK2 known?
Yeah.
It is.
Parts of it, I don't, is the structure of, I don't know, so.
It is known.
Yeah.
They're absolutely huge, and they're like positions on it that are not known. But they do have partial structures of like certain binding sites. It's kind of like the random interceptor. It's just really, really big and difficult to crystallize.
Yeah. I'm sure that there are disordered parts of it that are hard to get, and it's those often that are involved in regulation too. So there are, and this is sort of outside of my comfort zone. I mean, I've worked with these models. We've done some, but I don't remember all the biochemical details off the top of my head because it's not what I work with on a daily basis. This is sort of emblematic. I mean, this could be any. Except for the fact that CAMK2 forms these dodecamers, which makes it really truly pathological in terms of this exponential complexity. This is typical of we'll see plenty of examples of signaling proteins where they have multiple sites. Post-translational phosphorylation is just one. There are lots of other means of regulation of proteins by post-translational modification. We're only, because of the recent uptake in the amount of mass spectrometry that's done to analyze signaling systems, we're starting to get a handle truly on how common those post-translational modification states are. So there's actually a lot more multiplicative complexity here. Those things are all acting to regulate these molecules in complex ways. And the effect is to make very dense networks of these interacting partners. And we're really trying to get at the biophysical mechanisms by which they can control activity. So the standard thing that you do when you make a model of a biochemical system, you say, OK, I'm going to consider a few receptor components. I'm going to consider what activates those receptors and maybe a few downstream targets. So here I pull an example from our work on immunoreceptor signaling, where we have receptors that are activated by the binding of antibodies or by the cross-linking of antibodies that typically bind to antigen molecules, bind to other proteins or typically proteins that have specific, that are recognized by specific binding sites on the tips of these antibodies here, the so-called FAB arms of the antibodies. Here we're modeling a system where we actually trigger the receptors by cross-linking antibodies and making dimers of receptors. But here we have just, on an abstract way, just a ligand molecule that comes down, binds to a receptor. The receptor has multiple domains that contain phosphorylation sites. They're represented here schematically by bumps. The phosphorylation of these residues allows for the interaction with additional signaling molecules, which happen to be, in this case, kinases, so they're going to phosphorylate other substrates. So in a typical model, we say, OK, we're going to consider these molecules. We're going to consider these kinds of interactions between them, binding interactions, phosphorylation, so or maybe thought of as some change in state or activity of proteins, interactions that are mediated here, and then additional binding reactions. So what do we want to model? And then the next step would be to figure out what are the possible complexes of this system, what are the possible different states that can arise. And those would be the different chemical species is one terminology. And that's represented here by the vector x, which is a vector of the species concentration. So all the different possible species that can arise in my system, we're going to model them, their concentrations as a function of time. This is just a standard kinetic equation, kinetic rate equations for modeling a reaction network over time. Okay, so these are the species concentrations. These are the rates of reactions, and this is the stoichiometry matrix that determines for a given reaction how many species, which species are produced and consumed. The details of this aren't important. I'm sure you've all seen simple mass action kinetics. So we might have an example of a ligand binding to a receptor to form a ligand-receptor complex. So the species would be these, the ligand, the receptor, and the complex. So this would be an example of where a simple reaction network which has three variables. So we could model this simply as a three-variable differential equation where maybe I have that on my next slide. Yes, good. I often find that I think that I've forgotten to put something on my slides, and then it turns out to be there. So I'm jumping ahead. So, but we'll go ahead then, and I'll put this. You can see what I'm doing up there. So basically, all I'm saying is that we can model the time rate of change of any of these variables due to the action of the two. There are two reactions in the system. There's the binding of the ligand and receptor to form the complex. So that happens at a rate k times the concentration of ligand times the concentration of receptor if we're following mass action. And the term that comes from our stoichiometry matrix in this case is a negative one. The ligand is consumed by the firing of this reaction. And then there's a positive term that comes from the reverse, the dissociation reaction. So there's going to be a k. Did I call it -1? I didn't call it anything there. call it k minus 1 times the concentration of LR. And we can similarly write differential equations for the other species in this system. Is this familiar ground or something? Does anyone find not seen this before? Okay, good. So the idea is that when we want to do modeling, we have to find out what are the equations for this system. And that's going to be the first step. And then we need to solve those equations. And typically, if we have more than a few, solving them is going to be doing some kind of numerical simulation. Question? What, sorry? B. Good question. That's Brownian dynamics. Okay. So right. So good. Jumping ahead, then we need parameters for the models. We need the rate concentrations, initial concentrations really of the species, and the rate constants. And we all know this because we've been worked on developing simple M-cell models yesterday. And a key point is that given the same reaction network topology and even the same reaction network kinetics, we can simulate that reaction network in different ways. And one of the things that in the development of rule-based systems was that we wanted to separate the mathematical description of the network in terms of differential equations into a higher level abstraction that allowed us to fully take advantage of the fact that the same reaction network can be simulated in many different ways and has many different sort of semantics of operation. So the standard one is this differential equation. So normally we translate models into differential equations. That same set of differential equations, same reaction network, can also be simulated, assuming that instead of wanting to treat the variables as continuous over time, we treat them as discrete. And we have to modify our equations in that case, and in order to do such simulations, use typically something like the Gillespie's stochastic simulation algorithm. And that just gives us, instead of continuous variables over time, we can look at the stochastic fluctuation. So each realization of the stochastic simulation gives us a trajectory where there's some noise on the concentrations, and we have to, say, average many together to get back to the average behavior. But often the noisy system exhibits novel behaviors. And then similarly, we can take the same reaction network and simulate it, include spatial effects, and model them as, for example, partial differential equations or particle-based simulations, basically. OK. So we talked about this. So this is what it scales up to. So this is an example. I didn't put the picture in here. So this is an example of a signaling network that occurs frequently in mammalian cell biology. The epidermal growth factor signaling network actually has earlier origins than that. But EGFR is a particularly important component of many cancers. Mutations involving this pathway are highly overrepresented in cancer. And so it's been a subject of a lot of modeling efforts going back now a couple of decades. And so typically what one wants to do, the input to the system is here. The ligand is the EGF. The receptor is the receptor here. It acts through dimerization. The receptor becomes phosphorylated, and it can bind a whole series of different adapter molecules that determine these different branches. And one can generate data on the time courses of various complexes and various phosphorylation states of the molecules in the system. And so to develop a model of that, one considers all the different possible transformations. It turns out you can write down these 22 differential equation, 25 reaction network model of the system. So it's already getting to be pretty complicated at this level, complicated to keep track of. But there's actually a lot more complexity hidden under the surface. So here's a more schematic representation of the system. Here's the ligand EGF, the receptor EGFR. There are, and if those of you who know something about the system realize that even this is a highly simplified representation. There's a what was the courtesy of the operating of the I don't even know why. They're really pushing this. I'm not sure why. Okay, that was very obnoxious. Yeah, it's really strange because I'm still in full screen mode, but it somehow just got interrupted from that. Now it should come back. All right, good. I'm not even sure how I could prevent that from happening because it didn't come from an application that was even open. Okay. So we're back to a network level diagram and these represent the different interacting molecules that were in that earlier reaction network model. But now I'm trying to look at schematically what do I have, and this is a little bit that we considered here are a few more interactions than were actually in that earlier model. We have ligand binding to the receptor. The ligand binding to the receptor here induces receptor-receptor interactions or dimerization. And then the dimerization activates the kinase, which can phosphorylate substrates on the other member of the complex. These are phosphorylation sites in the receptor. And these phosphorylation sites serve as binding sites for these other adapter molecules, Schick, notably Schick, GRAB2, and SOS, which binds to Schick, to GRAB2. And SOS is a guanine nucleotide exchange factor that's responsible for activating RAS, for example, which one of the primary downstream molecules, and propagating signals that lead to growth and proliferation of cells. So this network, we could call this a module of just a few interacting components. And so we can see that this is actually more complicated in terms of the number of interactions that are considered than the model that was shown here. You can see that there's Schick and GRAB2 and SOS in this system, but there are more binding sites shown here. So there's somewhat more complexity, but it's relatively simple to understand. So we have 5 proteins and 20 interactions. But if we were to now try to write down a model that included the possible states of this system, we would get 170,000 differential equations we would have to manage. So obviously, we're not going to write that down by hand. And even the Holodenko model, in essence, just considers all the phosphorylation sites as a single one and neglects some of the other aspects of oligomerization here, and they wind up with these 30 species. So with relaxing some of the assumptions that they made quickly leads to this explosion. And that's really the key motivating factor here. So based on what we know about the biochemistry, a few known facts, the 20 facts about the interactions of these proteins, which we can express, and we can go through the literature and find those, basically put us at a no-go point in terms of being able to model that system in the standard way. That motivates why we want rule-based modeling, some way to describe these systems, to describe a model in terms of the interactions on this diagram, and let a computer figure out what all these possibilities are. Okay, so further examples of this complexity in signal transduction. And don't be fooled that this is somehow limited to, oh, this is just pathology of signaling pathways. This same thing happens, we already talked a little bit about CAM kinase 2 and the postsynaptic density, but proteomic studies of proteins in the postsynaptic density have shown that many of them have complexity that's comparable to this, what's shown here. So these are growth factor receptors, PDGF and EGF epithelial and basically growth factor receptors that are important in different contexts of signaling. But what they share in common is they have these disordered cytoplasmic tails of these transmembrane proteins, have lots of phosphorylation sites. Those phosphorylation sites bind lots of other effector downstream molecules conditionally upon phosphorylation. Typically, there are SH2 domains in these proteins that serve as these phosphotyrosine recognition elements. And there's a lot of overlap as well. So that a typical site typical phosphorylation site here in the cytoplasmic tail could bind several different partners. And I just did some simple bookkeeping here, assuming either 1 or 2 partners for each of these sites, and we get these sort of astronomical possibilities of complexity. So there's lots of different combinations of complexes involving these components that can and do form during signal transduction, unless somehow there are sort of magical rules to filter all of that complexity. So, some people believe that in fact this is obviously maybe an upper limit. In the case of a dimer, there might be 8 million possible dimeric species. You could imagine that different kinds of effects, cooperativity of binding of these proteins at different sites, would limit the number of states that are actually observed. So if for example, the binding of one protein to a particular phosphorylation site blocks the binding of proteins to nearby sites, that is going to reduce the number of states that are possible in the system. There's precious little evidence that happens. So it's an assumption that people like to make. You might convince yourself of, if you make some models, that you could have steric effects between proteins. But in the few cases where people have actually looked and tried to quantify these effects, they find that you can bind multiple adapter proteins per receptor. So this complexity is not going away that easily. Okay. Right. So I mentioned here, this is going back to this diagram. So I mentioned before, there's this, astronomical complexity in terms of the state space. However, the rules are simple to express and even to understand if you spend some time. So, but we need ways to handle this complexity in terms of if we want to set up a model. Okay, so one last piece of motivation here, which is going back to this multiscale concept. So I'm arguing here that rules are a nice way to concisely represent what's going on. And by rules, I just mean the edges on this diagram that connect the different sites of different proteins. And that enables us to connect a structural view of proteins in terms of the different functional domains, be they binding domains or catalytic domains. This is a protein RAS, which happens to be small. Its structure is well known. The function of basically every amino acid in this simple polypeptide are pretty well understood. And so at the molecular level and at the atomically resolved level, what we'd like to be able to do is to take our understanding at this level and use that to build network models of signal transduction. But as I mentioned, there's this kind of gap. If we start to write down the interactions of these molecules with other proteins in the network, we hit this combinatorial problem. So here's a network, and RAS is, I know where it is. It's right there. Or maybe I have it zoomed in on the next. Nope. So it's functioning in some very large network context. It has a number of different partners. This is very schematic. It has more than this. And the idea of rule-based is to bridge this, is to provide an abstraction that sort of bridges these two pictures. RAS in a network context interacting with other proteins. Now we're going to zoom in on the different functional components of the RAS protein, components that interact with other proteins such as RAF or the EGFR receptor, or that interact with the guanine nucleotides GTP or GDP, and so forth. So we can model that as in a sense as a state machine, and couple this to other similarly complex state machines, or even state machines of greater complexity in this whole network, which is shown here. So this is a schematic representation of a rule-based model called a contact map. It looks a lot like the diagram I have showed on the previous slides, where the edges just represent possible binding interactions between proteins, and the detailed names here aren't important. And each protein has potentially multiple functional elements that couple to other proteins. So in this view, the rule-based model is can be thought of as a structured, as a graph. Can be a hypergraph or a hierarchical graph, depending on the ways that one represents this. And this has a direct correspondence to an underlying set of what we call rules that describe the biochemistry of the system and in a fairly concise way. So there's more or less a one-to-one mapping between edges and rules and between the purple states and rules, which we'll see in the examples. Okay. And the key point is that this really is essentially a direct translation of what biologists who study signal transduction are actually measuring directly: interactions of different protein domains with other protein domains and other functional domains and other proteins, post-translational modification sites like tyrosines that are phosphorylated by kinases, kinase domains that interact with multiple substrates. And all of those can be concisely represented in the form of rules. So this sort of middle layer allows us to connect knowledge of structure to signaling in a network context. That's the key point. Okay, so how does this work? How do we actually go about describing the rules for a biochemical system? So at the most basic level, you have a rule that describes binding between two proteins. And the only thing that a rule adds compared to standard mass action kinetic description is a description of how the two proteins are interacting. So we have two proteins, A and B. They're going to form a bond. And they have here cognate binding sites. So the convention that we often adopt in the absence of further information is that the protein A has a binding site, cognate binding site for protein B, and we call it little b, and vice versa. And so this rule just says A binds B through these cognate binding sites. And the idea is that in its simplest form, the rule just says that a bond can form between these two proteins if they have a free binding site of each type. So an A molecule can bind to a B molecule as long as its A site as long as its B site on the A and the A site on the B are not bound to something else. So if they're bound to something else, that would block the binding. So this is a simple rule that selects all of these possible different molecules of A and all these possible different molecules of B. And what the rule says is we only care about this very local context of this binding site being free. I don't care about neighboring binding sites, whether they're bound to other things, whether they're phosphorylated or unphosphorylated. All of that doesn't matter. So you can think of each pattern in the rule, the pattern specifying the A molecule and the pattern specifying the B molecule, as selectors or wildcards, if you like. This A pattern matches all of these different possible particular instances of the A molecule. So if the A molecule is phosphorylated and not bound to something, not bound to the ligand, if the A molecule is unphosphorylated, bound to ligand, it can be phosphorylated and so forth. And we could have many other states of A. The rule only cares about the binding site. And similarly for B here. Okay? Here we don't know. Here all we're saying is basically we have like a Lego-like rule. We say that these two things can interact in this way. I haven't said whether this guy could interact with somebody else. I could have a rule that allows that to happen. So I could have a rule that allows A to bind to another molecule C at this B site, and then that would represent a competitive inhibitor of this B site binding. Okay. And I'm not constrained to only have rules of this. This is the simplest kind of rule. I can put additional constraints on both the A and B molecules to participate in this binding as I like. Okay. But in its simplest form, this just represents the most basic, and if we're truthful about our knowledge of most biochemical systems, often we don't know any more than this. We know that two proteins co-IP, and that's it, right? So we don't know the effect of any other modifications here on that interaction. We would like to know, perhaps, but if we want to write down the most simple model that describes that, this is maybe the best we can do in many cases. Okay. So we can make rules more complicated. So the part that I described so far is this, what we call the reaction center, the part of the rule that says what's changing when I have the reaction, when a reaction takes place. So here, what's changing is I'm adding a bond between two components of these molecules. Now I can put additional conditions on that binding interaction. For example, I could require that a ligand molecule is bound to an adjacent site on A in order for this binding to happen. I could require that the A is also unphosphorylated or phosphorylated. I could require, for example, also that this site be phosphorylated in order for binding to occur. So the idea here, and just to back up for just 1 minute, so the key thing is that each pattern in this rule, the A pattern and the B pattern, are selecting multiple different potential instances of each type. And all of those combinations are possible. So this particular rule that says A binds to B could generate lots of different AB binding reactions. Okay, and because there are no other restrictions, this is sort of the most general rule for this kind of an event. It's going to generate the maximal number of possible reactions between A and B. If I put additional restrictions on by putting more context, then some possibilities are now going to be excluded. I'm not going to select A molecules that are phosphorylated here, and so those are eliminated from my set, so I'm going to have fewer reactions. So here's the an example. I did, yeah. Imagine here now that I put this additional context U on these molecules, so that we only select these two out of the full possible set we had before. So this represents addition of context, and what I'm trying to demonstrate here is that in many cases, what's actually happening when I make a simplified reaction network is that if I model the AB complex as only one species, I'm basically saying I'm only considering a restricted subset of the possible complexes that can form. Right here, yeah. And that's a more subtle point, but the key thing is that when we go back to a model like the original EGFR network model here, hidden within this already pretty complicated system are several assumptions that have been made about what things can happen to this receptor complex that really limit the complexity. So one of the key assumptions that's made here is that the receptor gets phosphorylated. There are actually 3 different proteins that can bind to the receptor in a phosphorylated state, but it's assumed that those are mutually exclusive. So if you bind one protein to the receptor complex, and this is actually a dimer, even though it's called RP, it's actually 2 receptors in that complex. So the model is assuming kind of implicitly that you can only bind one thing, and that they're exclusive pathways. And so that assumption greatly reduces the combinatorial space of species in the system, but it makes my model, it sort of builds in assumptions that may make the model less predictive in certain cases. For example, if I want to model the effect of overexpressing one of the proteins in the system on the signaling, if I assume that all the binding is competitive, that's going to give me a different answer than if I assume that the binding is not competitive. Okay. So here's an example of a phosphorylation reaction. So I talked about binding before. So the other main category of reactions that one needs to consider in early steps in signal transduction are post-translational modifications, adding here a phosphate to a tyrosine residue on this A molecule. And I added here the context that the ligand molecule is required to be bound in order for this phosphorylation to occur, typical kind of signaling process. And here's the diagram again, showing that, okay, so we can build a network up over a series of rules. I'm not sure what I had in mind to put by putting that back. But if we go back, so now, if we go back to our modeling protocol, this was the first step we had before. This is what we want to consider in our model, the biochemistry. Now we can, the next step really, instead of trying to figure out what all the possible equations are here, we can simply write down what are the different molecules we want to consider, and then how are they wired together. So we're going to translate our proteins into these structured molecules that have different possible sites. And this is an introduction to the actual syntax of the BioNetGen language, which you'll learn a little bit of today. So this is how one writes the molecules. And then the biochemical steps, the rules, are represented in this form. And here I've shown a phosphorylation reaction. Again, so the receptor is getting phosphorylated on a particular site B, and the phosphorylation status is going from U to P. But there's a lot of additional context. So in this phosphorylation reaction, there's a kinase that's associated with the receptor that's phosphorylating an adjacent receptor in a complex. And it's representing all of that contextuality here is a little bit complicated. So I have my receptor that's the substrate. It's bound to another receptor that's in turn bound to the kinase that's doing the phosphorylation. So that's an example of one of the more complicated rules you might have in such a system. And then the rest, this part is the same. We still have the same rate parameters. And it's worth noting that even though we might have a very dense network of different interactions, the number of rules is the same as what we would have if we were translating this into differential equations. It's just that each rule now generates many different reactions. So typically, if we're going to consider a model that we had a standard reaction network model, essentially the number of parameters for the two kinds of approaches is more or less the same. They have a lot more reactions because of this combinatorial complexity, but we don't have lots of additional parameters. And then we can simulate and analyze in different ways. And BioNetGen was primarily developed as a compartmental or single compartment modeling tool to use ODEs and stochastic simulations. And as part of the BTRR, the MMBios project, has been to develop ways to export BioNetGen models into MCell to do particle-based spatial simulations. And Jose and Devin will be showing that this afternoon. Okay, let me see. Bear with me for 1 sec. I just want to see something. Just trying to figure out the pacing, because we should take a break sometime reasonably soon, since I've been talking for a while. 10. I know I'm going to break by 10 is my target at the moment. I'm just trying to see what I want to show. Okay. All right, good. So let me just say that to finish this component by saying that overall, a rule-based model looks, at least in part, the key ingredients of the rule-based model are these two things. The molecules, which define the different proteins or other signaling components that I have in my system in some structured way. So I can represent the binding sites and the post-translational modification sites on my molecules. And then I write rules that modify those interactions. They create bonds, or they carry out phosphorylation. And so, these are snippets of this BioNetGen modeling language that allow us to describe these interactions. And as I said, we'll be talking in more detail about how we actually construct those models and giving some examples. But this is the essence. And by essentially, one can build up very complicated, large-scale models of signal transduction systems in a structured way, and we can visualize these networks and we can simulate these networks. Sort of over a very wide range of possible complexity. So, yeah, I wanted to say, so are there questions about that?
So, I just kind of like, I mean, if you take like a Hodgkin-Huxley type model, and if you have all these differential equations, and say if you translate it to this BioNet Gen language,
Mm-hmm.
then you're saying from there you could go to M-cell kind of representation?
Yeah. Now, Hodgkin-Huxley, so the one caveat about that would be that so far all I've told you about rules, the rules that I've given you so far are described by mass action, elementary mass action. It's difficult at the moment to specify, or I should say it's easy in BioNetGen to specify non-mass action kinetics. We can specify arbitrary rate law functions in BioNetGen. But there's no simple way to translate that into spatial modeling domain, because within M-cell, a bimolecular reaction is really a local event between two molecules. There's no direct way to make that rate depend on some global property of the system. From an algorithmic point of view, that's not, and a programming point of view, that's not complicated. But from a biophysical point of view, it's a little bit problematic. So.
I see. So the rate constants themselves will not be right.
Yeah, you need to. I mean, if you wanted to transform a model that has non-mass action in it into M-cell, you have to figure out a way to break down those break down that mechanism into its mass action components. At the moment, that's how it would work. So, yeah, that's the one caveat. We would really like to be able to couple to handle non-mass action, as I said, in the simulator. But then you have to think very carefully about what that means from a biophysical point.
What your system is technically, I mean, it doesn't know whether it's a local species or whether it's a global species in your bigger system. So
I don't know what you mean by a global.
If
Yeah.
you take like a Hodgkin-Huxley formulation, and if I plug it in there, I mean, I can see that I can write equivalent structure or equivalent code.
Mm-hmm.
But then when I'm trying to, I mean, I see what you're saying, but when I'm
In M-cell, there's no global variable. There's no V, for example, that spreads over a whole region. I mean, it's not that it couldn't be, but it's right. So correct me if I'm wrong, Marcus, if I say anything. But you can't. But at the moment, there's no notion of a collective property. I mean, there are outputs, but those outputs, those collective things. So you could measure the total amount of phosphorylation in the system, right? In a signal transduction system, you could imagine that the total amount of phosphorylation feeds back by that represent it could represent a substrate for, say, phosphatases. They could feed back on the system by saturating phosphatases. But we don't have there's no clear way to represent that kind of process in a spatial context. Yeah.
Things like field, that
Yeah.
would be something that could be quite natural in the spatial community. It has a spatial aspect to it, a field, whatever, electric field, hydrodynamic. I mean, some property that changes, as a function of space. And then there's other aspects that are non-mass action that, in a spatial context, if you think about molecules that just are not, doesn't almost make sense to I mean, you don't know what they actually mean once you think about particles. do you think like field, or, I mean, that could be done, and I think it's something you're thinking that may make sense, that you have something that's the property of the system that kind of influences the particles, but you know, we have to think about how does an electric field influence diffusion, what kind of properties do you need to give particles. So I mean, there's lots of, once you start thinking along that route, and there's lots of things you need to both think about conceptually and actually go to the math and kind of figure out how does that actually work. Do we know how to solve free diffusion? Stuff. Certain things you can still do, but
Yeah, I mean, if you want, if people are interested, I can talk about why non-mass action is a problem, and it just, but I think that maybe that's a little specialized, so we can. I'd be happy to return to that subject, though, if you have questions. We can work. If you think about how, you know, in a way, and just an exercise I would encourage you to think about is how would, you know, what does Michaelis-Menten mean in a spatial context? How would you model a Michaelis-Menten reaction in a tool like MCell, where you have a bimolecular event is the driving factor? So it's tricky. It's quite tricky to think about that. Any other questions?
So, I mean, at the end of BioNet Gen, you will get a set of equations which we simply gonna type into MCell, or are they incorporated into each other?
You can you can directly, so you can take, if you write your BioNet Gen model in correct format, and there are some restrictions. Clearly, the most important being you have to write mass action, a mass action-based model. But if you do that, then we have developed tools that allow you to import that model directly into BioNet Gen. So you can, and I have a picture. There are two basic paths for that. Let me find my picture. Thank you for asking that question. So yeah, to give you a picture. So you can, we can write, this isn't a full diagram in some sense. If you write your model in this compartmental BioNet Gen language, which we're going to talk about, you can import that directly into CellBlender. You can also take your BioNet Gen model, export it as an SBML model. This is a common interchange format that many different modeling tools accept. And there's a way to import that also directly into CellBlender. And we're primarily going to show this component of the interface today, but we have the this import capability is built also directly into CellBlender, and we have a tutorial that shows you how to use that as well.
CBNGL, does that have orientation like MCell does?
It does not, but the translated model does handle that, making certain assumptions. So there are a few things that are tricky to do. There are a few kinds of transport that it can be done in CBNGL, but you have to do it in a slightly different way than you would typically do in MCell. And if you do that, then you could translate the model and everything would be fine. But it does not use the orientation per se. But it sort of BioNet Gen understands how to translate the model correctly to handle basic cases. If I say any more, I'll be wrong because it's a very confusing topic. But I'm confident that we've handled it correctly because we thought a lot about how to deal with it. Yeah.
We could define reactions directly in CellBlender.
Yes.
So importing from CBNGL, how does it benefit?
If you have like so the example, one of the examples that we do with the tutorial involve, and I think the network that Jose and Devin shows what has 28. How many species do you have? 70, 80 species. So 80 species and how many reactions?
350.
So if you would like to type 350 reactions by hand, you can do that. The problem is not so much whether you have the stamina to do it, but whether you are good enough to do that without mistakes. And if you happen to make a mistake, would you be able to find that mistake in a reasonable amount of time. And I mean, one of the reasons that when we developed the BioNetGen for this immunoreceptor signaling system that I talked about, this particular, I usually give the numbers, but this particular system, do I have it somewhere here? This particular system, so just these 4 components and these, I think 8 rules. 1, 2, 3, 4, 5, 6, 7, 8, 9 rules. That's 354 species and 3,680 reactions, which you can bring into Cell Blender pretty easily. We simulate this system in Cell Blender, but with an MCell. We developed this because originally, when I was part of a group at Los Alamos, we were developing this model. We literally did it by committee. We had 6 people, and each person was assigned 50 or 60 equations, and nobody could do it without making sign errors or factor of 2 errors. And you get into heated arguments about there's symmetry issues as well that come up here. So and then that's just one structure, and you say, okay, well, but there are actually 2 phosphorylation sites. Forget it. So the idea is that at some point, which I think is at a number between say 20 and 50 species, doing it by hand becomes just a really bad idea. And even at that level, I mean, you can do it, you can write it for yourself, but then how are you going to, somebody else in your group comes along and wants to use that model, how long is it going to take them to figure out what you did and what assumptions were there? And so one of the big advantages of rules is you're directly translating this information into a form that's pretty easily understood. But what this gets, what any of these interactions get translated to, maybe typically these are 50 reactions each. So there would be 50 reactions describing these different phosphorylation steps sprinkled throughout your network. I mean, maybe you're good at you group them together, but it gets really, really hard to keep track of these things. So I mean, I think one of the real advantages of using this a very descriptive system that breaks up my proteins and my biochemistry into explicit rules is that all of the assumptions then are made quite clearly, as opposed to getting buried into this combinatorial snowball.
I don't know if I'm the only one, but I still don't understand how you can distill so many molecules and reactions into so many rules.
Right. That's a very good question. So how does that work? So let's go back to our. And I'm sure you're not the only one. This topic is very confusing, actually. So it's really here that the idea is that my rule just says A and B bind together. And the idea is there could be many, many different species that contain A or B in my system. There could be ones that contain both A and B in my system. And the key idea is that a rule just says that these guys have binding sites that recognize each other, and I should find all places in my system where that can happen and make the associated bonds. So here there are, I've shown 4 different forms, 4 different individual species that can satisfy the reactant pattern here for a free A molecule, and the reactant pattern here. And there are another 4 that match here for B. But imagine in some system where I can have complexes with A and B, there might be hundreds on each side. And every single one of those is a potential combined. So there would be, 100 times 100.
So do you just specify like A, B must correlate with site 1, A, B, A combines with a ligand, and then it generates all the possibilities?
Yeah, exactly. That's what the rule is. So here in this diagram, and I'm trying to think of, I'm just trying to look at what's in terms of context. So here, basically, each rule, there would be a rule for each phosphorylation site, for example. And it'll probably help when we go through when I actually just walk you through a couple of rules and you see how it works and how it builds up. But I'd have a rule for each of these guys, and then it would, but there are different because each of these sites can be independently phosphorylated. There are many, many different states where you've got a phosphorylation here, and this site is not bound yet, and then it can bind to GRAB2, for example. This one only binds GRAB2. There are no sites that bind to. I see. Yeah, they're not even overlapping. So each of these sites then, when it's phosphorylated, can bind either to SHIC or GRAB2, depending on which one it is. And the rules for those just have very local context. So here, there are only, I forgot how many I said. There are of order 20 rules for this system, because they're only saying what the local requirements are. And in order to get kind of in the reaction network modeling world, and Jose's really done some nice work on this problem. In the reaction network modeling world, what happens is, one way to think about what's going on is that life gets much, much easier if you say that there's only one instance of this reaction. I'm only going to, the protein gets phosphorylated, and then it can bind to, pick an adapter, instead of, and I'm not going to worry about all the other possible states that protein could take on. So, in a way you can think of it as putting additional context on these rules. That's what this picture was supposed to illustrate. That in the reaction network world, what happens is you just say, well, I'm only going to worry about A binding to B when both of them are unphosphorylated, because I don't want to deal with. If I deal with the possibility that it could be phosphorylated or not, then I'm going to have 4 reactions. But if I just require them to only be unphosphorylated, then I have 1 reaction. So that's typically the assumption that's being made. So if you deconstruct a typical network model that we have done for hundreds of examples, you get additional context. You'd say that when I say additional context, I mean in the rule you say that the A site, this other site must be unphosphorylated on A, and this other site must be unphosphorylated on B. And what and the question is that do we know that, or we don't know that? Is the problem. If we did, we could just write it in our rule. Okay. Was that helpful?
So by putting in these constraints, you're removing certain reactions, and
Yeah.
is that how you're
Yeah.
simplifying it?
So the way I think about it is that the BioNetGen is modeling reaction sites instead of reactions, and then the context under which those reaction sites are allowed to interact.
Right. Yep. Exactly. So here I haven't said anything about the context. I've just allowed all contexts. I just require that there's a free site on each molecule.
In the middle of that.
And they could drag along a huge snowball of other things and still bind. At some level, that's going to become unrealistic. At some level, other things that happen to my protein are going to affect whether these guys can bind.
If you're modeling the traditional way, you'd have to write out every case in which they could bind.
Right.
as a separate
Or just
reaction.
limit them to one.
Right. But in this case,
Another way to think of it is you have, if you have your network of species, and you have a reaction network, in the standard approach, you just say, maybe this is A, B, and C, and they're somehow binding to each other to form a complex. You say that A, B bind first, and then and form a complex, and then these guys bind and form another complex. But you allow only one ordering. You don't allow this bond to happen and then this bond, for example. And so that helps you to simplify possibilities in this network, as opposed to just saying they can both bind and all possible sequences are going to be considered.
But aren't we losing some information by eliminating those reactions?
We aren't eliminating these. I mean, when you do this, you're saying, like, if you have, so let me just make an explicit model. So you have a B, and let's say B can bind A, and it can bind C. So in the rule-based version, if there's no interaction between these sites, I can have an AB complex, a BC complex, or an ABC complex. in a non-rule-based version, I might say, okay, in order to get rid of some complexity, I might say A binds B, and then B binds C. And then I'm missing. I'll have, in that case, I would have an AB complex and an ABC complex, but no BC complex. So I got rid of a state. Here, in the binary case, it's not so much. Up to 3, it's not so much. You go one more beyond that, that's going to start to amount to a lot of states. But does that make sense? So in the conventional approach, I would just it starts to get really tedious if I have to write down A, B, A, ABC, and BC, for example. So I might just choose to ignore this one. Put two modifications here and two modifications here, and this is starting to become a large number of things now that I'm getting rid of by not considering this complex. And there are plenty of times when this is perfectly justified to do. Maybe it's known biologically that BC is just not observed. You can't co-IP them or something like that. That might not be hard proof, but that's suggestive that maybe I can make this assumption. But more often than not, we just don't know. And I think the rule-based approach, what it makes you think about these possibilities. It make you think about complexes that you might have otherwise wanted not to deal with. And once in a while, something pops up. It turns out that actually, BC is really important. I went to a talk on Sunday where that was talking about EGFR signaling, and literally one of the complexes that would be kind of a throwaway thing, that something, well, this can happen. You have to have a good picture. You have your EGFR molecule, and it binds to another protein called GAB, for example, in this case, which binds to another protein. Doesn't matter what they're called. And you think, well, okay, it's true that this bond can break, and you could have this complex of these two. But maybe that's not so important. I'll just throw that at a system. And it turns out that the whole mechanism depends on this dissociation and this seemingly kind of throwaway complex diffusing through the cytoplasm and having its activity maintained by another protein. So my point is just that in thinking about these different complexes, there's a lot of potential information. Yeah.
You can probably speak for that much better than I can, but I think one attractive thing to me beyond just that it's a really nice interaction that really helps you understand very complicated systems at a higher level. You don't have to think about all the details, you just think about what's there, what can happen, and what really happens is a completely different question. But in addition to that, also from a simulation point of view, once you have millions of possible things that could happen, not all of them happen.
Yeah.
Some of the species that occur may actually never exist in the real system. So having this kind of abstraction also helps you, and I think this software does it already, but you don't have to actually look at all the reactions. You don't have to look at the ones that actually happen. So if some of the species never occur during a simulation, those reactions are useless. You don't need to write them down.
Yeah.
So you can come up with ways to just simulate whatever is happening. Only the species that occur in the system will actually be considered. So that's not what we're doing now with this translator. We're really generating all the reactions. But you could imagine that if you have a very complicated system, maybe only 5% of all the possible reactions ever happen. So it makes a lot of sense only ever consider those. The rest is just noise, and actually to generate them. The possibilities that might occur. So I think this abstraction really allows you to do that in a way that you could never do if you really just write everything down. Because once you write everything down, you have to write it down.
Yeah.
You don't even know if that reaction ever happens, but you've got to write it down anyway. Whereas with other folks, you don't have to.
Yeah, so that's a great segue. Let me just, this is a short topic, and then we'll take a break, because I'm sure everyone would love to stretch their legs. So the basic idea here, just to amplify on Marcus's point, we talked a little bit about a BioNetGen model is comprised of a set of objects defined by molecules and rules that define their interactions, and we can conceptualize this in terms of that nice little interaction map. You have proteins with different sites, and those sites are connected to sites on other proteins, and either through catalytic transformations or through binding. So that's my biochemical description. That's rewirable. I mean, as I go through and I read the literature and I find out there's another interaction, I can add that to my diagram. I can add a rule for that. I can investigate different possible sets of rules. Maybe group A believes that this particular interaction is important, and group B says we never observed that interaction. I can have alternate models with different hypotheses in them. Then I take those different models. I can basically pass that model to BioNetGen, regardless of how much complexity is here. There's a caveat, but I'll get to that in a minute. I can then make my reaction network and run a simulation. And I could say that this reaction network is an exportable thing. I can pass this to MCell, for example, through Cell Blender, or just pass it directly to MCell and do a simulation, a spatial one. Here I'm showing well-mixed simulations, ODE and Gillespie, and then we can do analysis of that. So we can investigate many different structures here. We don't have to worry about the size of this reaction network directly. And so that's a powerful capability. And as Marcus said, I mean, you're not committed to one form or another. You can change that. So let me just walk through what does this mean, network generation. So the basic idea is I've defined my set of molecules that are relevant to a particular signaling pathway. And these circles represent different components on those molecules, and these are the different names. These are a receptor and a couple of kinases. Okay, so and these define what we call in BioNetGen the seed species, or really the initial species that are present in the system. Another way to think about these is these are ground state molecules. They haven't bound to anything, and they're not modified in any way yet. And so in a basal cell, our picture of a cell sitting there at rest is that all of these are not active or bound. That's not true, because in the basal cell all kinds of stuff is happening, but we'll forget about that for now. And so, okay, so these represent the starting point. I have a network that has 4 species in it, and now I've defined a set of rules that say how those 4 species can interact with each other. I apply these rules to these species. So when the initial thing that can happen, maybe Lin can bind to the receptor, and this the ligand molecule can bind to the receptor. So those generate a couple of reactions, and we'll go through in a little bit more detail after the break how that works. And so those possibilities are investigated, and then new reactions are generated from those from the rules acting on species. And at each step, some new things will happen. So in the first step, ligand binds to receptor, and receptor binds to Lin. And I happen to know this. I haven't shown you the rules here. Okay, so there are 2 reactions that are generated, but those reactions themselves generate new species, and those new species now have new possibilities to react. And as I said, I'll walk through. I have examples in more detail, and those get fed back into the network, and we just keep iterating. And at some point, if polymerization isn't happening, if you can't form infinite species, this is going to converge, and you'll get from these four initial species, 354 possible species and 3,680 reactions. And both the standard tools, the standard ways of simulating reaction network models require that I determine what these are in advance. That's what Marcus was alluding to. And these numbers, so this is just kind of the tip of the iceberg kinds of numbers. You can get much bigger reaction networks, and often, in a given simulation, you might not realize many of these species, and some of the reactions may occur at very low rates. So there's something not exactly optimal about this process of having to generate in advance what all the possibilities are. But nevertheless, if I simulate with differential equations, it's very hard not to do that. You need to know what the equations are before you can solve them. So one way that we've developed to get around this problem is to, instead of simulating a whole network and all the possible states, we make an instance of that network. We basically we say that initially we have a certain set of molecules in our system that are, you could think of them as particles, like in an M-cell simulation, or you could just think of them as particles, but they have no coordinates in a box here. And basically, at each step, we apply the rules to determine what things can react with what, and we pick one of those events to advance our simulation to the next step. So the idea here is we're combining a discrete event simulation with an instantiation of our molecular network. And for those of you who do structural simulations, this is the most natural thing. If you want to simulate a protein solvated by water, you don't simulate every possible conformational state of the protein and water interactions all at one time. You just pick a particular instance, a particular protein solvated by some water molecules. You compute the force generated by that to advance you to the next step, or maybe you sample a configuration at the next step that's close to the one you started with. And here we're doing the same thing. We're saying we have a given set of signaling molecules. This is a little bit confusing in a sense, because this is already in the middle of the simulation. Complexes have formed, post-translational modifications have taken place, and so forth. But if you think of starting this out, initially I just start with, say, my ligand molecules and my receptor molecules and my kinase molecules. They're all unmodified. And then, because the ligand has been added, ligand starts binding and starts activating receptors. But the key idea is I'm not trying to generate all the possible configurations, but just the ones that happen because I have chemical reactions taking place in my system. So the idea is that by doing this kind of stochastic simulation, I can start with a network that has a simple set of rules that generates a relatively small network of, say, here, maybe a few hundred or a few thousand reactions. And over time, I can gradually increase the complexity. Maybe I know about new proteins that are part of that network. I learn about new interactions that can take place in that network. So my network is going to grow in size over time, typically. As I learn more biochemistry, I make my network more and more complicated. And what we wanted to do was develop a technique that, as we make the network more complicated, the simulation time remains roughly constant. And that's what this network-free mode of simulation allows you to do. So that's shown in blue here. If I run a simulation network-free, then as I make the network more and more complicated, more and more interactions, it doesn't really blow up on me. I'm able to continue to maintain a fairly rapid simulation of that system. Whereas if I have to generate the reaction network, like if I want to use differential equations to simulate my system, the network gets, as the network grows in size, the computational time increases exponentially, or it increases. Here, I think the slope is around 2, but it increases very fast, and very quickly, I run into a situation where I have too many reactions to even store in the memory of the computer. So there are all kinds of limitations that arise because of that. And what you can see is that there's a crossover point. So initially, there's some extra cost associated with simulating a particular set of molecules. So there's a cost to having to find the events at each step. I have to figure out what things can react to what. So there's bookkeeping that goes on there, and that's work, and that represents additional CPU time that needs to be expended. However, that cost doesn't really increase very greatly as I make the reaction network larger. And so when you get to a certain level of complexity, which is going to be problem specific, but it's typically on the order of hundreds to thousands of reactions, this method actually gets faster, and it makes feasible simulations that otherwise can't be done. So for example, if you wanted to simulate CAM kinase 2 that we talked about before with its 40 to the 12 states, we can actually run a simulation of that system fairly easily using this kind of kinetic Monte Carlo approach. Yeah. Question?
Account for the runtime reducing.
Oh, people love, yeah, that's a, because, yeah, this can happen. So how do you systematically scale the size of a network? So what we did here, what we did in making this case study was to try to make different kinds of changes to the structure, to the interactions between these proteins that would increase network size. But the kind of coupling between states that you get is not can vary as well as the network size. So if you think in terms of, if you're familiar with differential equation systems, if you think of the Jacobian of the system, what's happening here is that the Jacobian in this case is actually denser than the Jacobian here. This is really this one has a really sparse Jacobian, and so it can actually, you can, if you set the simulation up in the right way, you can take advantage of that, and it becomes more efficient. Even though there are actually more equations here, the structure of this system is just such that you can solve them more efficiently. And it's exactly why I can't tell you, but I know that it's related to how coupled the different species are to each other in the system. Yeah, that's very problem specific. Yeah, so yeah, it's probably bad. I mean, you know, this is an honest point. You know, you'd like to see it be going up here, but what happens is the next. This one is this one. So you could simulate the ODEs here, but you notice that there's no circle out here, and that's because you can't, because and I forgot why this is true, but for the stochastic simulation of this system, it exceeds the memory, the core memory of this computer, and he couldn't run the simulation. These are really typical kinds of problems, not, you know, that you encounter just practically when you're trying to make a more and more complex model. And you know, the details are going to vary, but you're always going to hit this point. And where you hit it, you know, it depends on what biochemistry you're considering, how ambitious you are, and so forth. So one of the goals we don't have, so now when we export a model from BioNetGen into MCell, we have to generate the network, and we have to export that as a reaction network into MCell. So an MCell can handle fairly large reaction networks, but it's gonna start to choke somewhere out here. In this region, I think it's gonna be not fun. Not to mention that if you have that many diffusing molecules undergoing biomolecular reactions, things are going to get slow too. But it's going to it's going to suffer from the same limitations. And so one of the goals of the project, which is not yet realized, is to bring these network-free kinds of simulation methods into MCell, so that and that requires MCell to basically be able to do a rule-based simulation. And we have algorithms designed for that, but it's just not been fully implemented yet.
So as it stands, it generates the whole network?
Yeah. Okay. So to take a BioNetGen model into MCell, you have to generate the network. So from a practical point of view, you know, models of some few thousand species and tens of thousands of reactions, probably the limit of what you can do. If you start to hit problems with that, you know, you should definitely, and if you go away and, you know, anyone has a problem where you feel like you really need network free, definitely get in touch with us, because we're looking for problems like that, and that can help us to accelerate our development process. I mean, we have the tools to do this, but it's, you know, having an application that seems relevant to drive it would be really helpful. Okay, let's take a break. It's a good time.